Welcome everyone. My name is Carl Larson from RMCG, and today at the cutting edge in advancements in biopesticide for profitable vegetable production. I'm a member of the Soil Wealth and Integrated Crop Protection team, and we're very lucky to be joined by a couple of great panelists and renowned international researchers to take us through some really excellent content today. So a little bit of an overview of what we're going to cover, starting with biopesticides and their application, and also some practical tips and tools around how to maximise their success, but also what are we seeing in terms of new products and emerging trends and some potential future innovations on the horizon there. And we've got some excellent perspectives from both the international and the Australian point of view, and the chance to have some Q&A as well. So a little bit about what the Soil Wealth and ICP project is, just very briefly. This is a research development and extension project that focuses on improving soil management and plant health, hence the link with biopesticides today. It's aimed at getting research to practice out on farm and vegetable growing enterprises around the country, working directly with the growers and also their advisors and agronomists. We delivered phase 1 from 2014 to 17 and are currently halfway through the second phase. And it's very much around responding to industry needs. And over the past 2 or 3 years, we've had quite a focus on biopesticides and alternative product use. And one of the questions we get continually asked is, how do you know what works and what's the real deal and what isn't? And so we've done a couple of different things around developing a biological products database as a quick reference tool for people to look up and find out and sort the wheat from the chaff, if you like, but also running events like today to really get those insights and build our knowledge collectively. So before we go any further, I might just do a quick little bit of housekeeping on GoToWebinar. No doubt, given the current circumstances, people have joined a lot of webinars, whether or not it's been through this particular platform. I'll just give you a quick overview to make sure you can get the most out of today. So the two things I just wanted to point to, one is the question pane on the control panel. Please feel free at any time to type in your questions. They'll come directly through to me and I can put them to our speakers. And also you'll notice there's a little handout drop-down box there, which has some of those resources that I've mentioned and other fact sheets for you just to click on and download and take away with you today, rather than having to copy down or take a screenshot of any long URLs that inevitably don't work. So hopefully that makes it easier for you as well. What I would like to do is just launch a very quick poll. So you'll see up on your screen now a question asking how much experience you've got with biopesticides in vegetable production. If you could select one of those 4 options, completely anonymous, just to give us a bit of a better idea of who's on the line and where you're up to. That just helps gauge interest rather than unmuting 50 odd people, because we had quite a few register for this session. I can see now we've got about 80% of us have voted on that. Just ticking over 90 now. So I'll close that off and share the results back with everyone. So looking at that, quite a strong cohort in the not much, around just under 50%. Over a third of us with a bit, and we've got some bookends there, 6 to 12% with lots and none but keen to know more. So quite a spread, but that just helps us and particularly our panelists just pitch the information correctly. So thank you very much for taking part in that. Okay, so I'd like to introduce our first speaker, Professor Travis Glare from Lincoln University in New Zealand. Now, Travis is the Professor of Applied Entomology and Director of the Bioprotection Research Centre at Lincoln University. He has an international reputation and has been published widely in the field of entomopathogenic microbes and the use of biopesticides. He's also worked on a range of international projects to develop novel biopesticides as well. So Travis, welcome. I'm going to share your screen so we can bring your presentation up. You can just click accept and that's up. So we can see that and the screen is working. So Travis, over to you. Thanks.
Right. Well, thanks, Carl, for the introduction. I'm never too sure whether having an international reputation is a good or a bad thing. It could be taken either way. So Carl's asked me to talk about biopesticides in sustainable vegetable production, but I'm going to go a little bit broader and where some of the trends are at the moment. And so in 15 minutes, I want to cover a bit of the sort of new areas we're looking at, things that really interest me. I've put on that first slide, there are no silver bullets. And this for me is really important because we have spent 30 years in biopesticides trying to find the chemical equivalent and trying to find something to replace chemicals. And it's the wrong way to go about things. So if you're looking for the single product that's going to solve all your problems, this is probably the wrong place for you. But multiple problems, multiple solutions is what we should be doing. Just got to work out how to change my slide. Here we go. So there's lots of drivers for change around why biopesticides are becoming so much more important these days. There's the societal ones, the environment and health, a lot of market push really for safer products. Regulatory, there's been a lot of withdrawal of chemical pesticides. There's residue issues on export crops. And the market's demanding less input. It's also the increasing costs of new chemicals to develop. And anyone who watches the industry will see there are fewer and fewer new chemical solutions coming out every year. So we need something. There's a number of bioprotectants that can be put into the sort of green category, and macroorganisms, whether it's ladybirds or parasitoids, the microorganisms, botanicals, plant extracts, some of those have some really good activity against pests, and semiochemicals for the insects. Today, I really want to talk mainly about the microbes, and I like to put up this little thing I call my wall of death. There are thousands of microbes that kill insects, and there are thousands of microbes that work against diseases, and even some that work against pest weeds. I'm an insect pathologist, so I tend to talk about insects first, but just most of what I say is applicable to the other categories of microbial-based biopesticides. So I'll use a roughly limited definition of biopesticides, where it's the active ingredient is a microbe or the bioactive from a microbe that is then formulated. And by formulating it, we're making what is really an analogy of a chemical pesticide, but based on a live organism or its derivative. We're also really just talking about one approach to using microbes, which is the inundative. So where you want to use it and spray or drill or drench in mass. So there's other ways to use these microbes, whether it's a point release or exotic introduction or even conservation biocontrol where you change the habitat. I'm going to concentrate very much on inundative, what we call biopesticides. I like this little definition that was done by a friend of mine, Wendy Gertler from the US, and she did the first three of these when we were having a workshop in 2012, and it's the eras of biopesticide development. So from about 1950 to 1980, there were the visionary entrepreneurs, the people who could see where these microbial-based products could have a niche and could be useful, and they would develop them through for very much a niche use, whether it was one insect in a crop where you just couldn't use a chemical or one spot where it worked very well. From about the 1980s, some of the big players got involved and decided they were going to make these things mainstream. And to some extent, they were visionary as well in that they could see where the market was going, but they were ahead of their time and the science hadn't kept up. And an awful lot of money was spent developing some relatively poor products, and all the big companies got rather scared of it and went back to their chemicals. So then from about 95 to really 2012, it continued with incremental gains on those visionary entrepreneur products. And it was just the science was getting better, the formulations were getting better, same sort of organisms that we were using, but just better products coming along. From 2012, this is what I call the second coming. And this is where the market's really taken off. And it's that perfect storm of chemical being less attractive to use, all being withdrawn, pest impacts are becoming greater because of things like climate change and also just the way we farm and the intensity of our farming. And the regulators and public are putting a lot more pressure on looking for green products. This manifested to start with on the left hand there, where the big players, and by that I mean Bayer or Syngenta or Monsanto, started buying up small niche biological microbial companies, and they paid some big money for what were really single or one or two product companies, and they were just their way of rapidly getting some microbial based products into their portfolios. This has continued. They're not quite paying those stupid prices anymore, but they are now trying to develop their own biopesticides in-house, and it's seeing the market increasing somewhere around 18% in revenue each year. And so there's some really impressive numbers over the last really 15 years now this has been going, and it hasn't slowed down. So you're starting to see some real market penetration by some of these biopesticides. So there are lots of successes. Some successes because it's still only making up about 7% of the pesticide market, but there are a lot of products in all sorts of different formulations, different organisms, and in different countries. Mainly bacteria and fungal species are used or their derivatives. Now, I'm not really going to go into much detail on the organisms, you'll be happy to hear. But what I want to talk more about is the concepts. I put this up because one of the biggest success stories in biopesticides from microbes is Bacillus Thuringiensis, and probably a number of you use this now in different ways. It has some real benefits. One is it's been a product since 1938, and so long use and safe use. It is really well understood. It's also a chemical equivalent, much more than most microbial-based biopesticides, in that it's based on a single toxin most of the time. So that means that growers could understand how they were going to use it because it didn't differ much to what they were already using. So there was a huge amount of uptake. Up to about 2005, it made up 90% of all biopesticide sales for insect control. Now it's gone down to probably around 65% now, as all the other products are starting to get some good uptake. It's also had the benefit of having strains that were active against caterpillars, the new ones discovered active against mosquitoes, the new ones are active against coleoptera. And so you could start to use it against different pests, and that was important for large sales. Of course, biopesticides for all growers have issues and have both perceived and real issues. And it depends on which organisms you're talking about. You know, are they virulent enough? Sometimes they're too slow to kill and the damage on the crop is as bad as if you didn't treat because they won't kill fast enough. You know, they're very susceptible to the environment in many cases. Complex life cycles. There's a perception that it's expensive and high production and research costs, which is not actually true when you compare with the developing a chemical costs about 10 times as much as developing a biological. But then on the other side, lack of profits. If you can't get into the big markets, how do you make enough to cover the research and development costs? Then there's the last one there is really where I think the biggest problem lies. It's the expectation of a chemical equivalent. You know, farmers and growers have been reared on using chemicals. I've got relatives who would love to have DDT back. It was effective, it was cheap, it just wasn't safe. And so that thinking has colored how we're trying to use a lot of these biologicals. And that's one of the things I want to talk about today. One of the interesting things that we're starting to find about a lot of these organisms is that they actually have multiple ecologies and they have multiple activities and multiple benefits, far more than what we've generally looked at. And so you develop a biopesticide for an insect, you spray it on a crop, you count the number of dead insects, and that's your activity. But what we're finding is that a lot of these actually interact with the plant. And the little picture down the bottom, trichoderma is a perfect example of a fungus that will give you, in many cases, enhanced plant growth. But it's not the only one. And a lot of the traditional long-used organisms that we use for insect control or plant disease control will also interact with the plant and give you enhanced plant growth, or increased soil macro and micronutrient uptake. Then you get these weird activities where you find a benefit against the thing that you weren't targeting. An insect pathogen on a plant can give you resistance to a plant disease. And these are things that we weren't looking for, or say drought tolerance. And this is what we really should be measuring. So what is the benefit to the plant and to your crop and to your final product, rather than just the thing that it was developed to control? Just one example of this is a lot of organisms go endophytic in plants. They actually go in and live inside the plants. Nice little photo on the right-hand side there, where this is the ryegrass endophyte that's developed in New Zealand. Almost all ryegrass seed is sold with an endophyte in it that controls against a lot of insect pests and also gives you some drought tolerance. Now that's an example of basically an obligate endophyte, a fungus that has to live inside the plant to have those. But we find a whole lot of the other things that we use, the other microbes, also like to go inside a plant. That gives us some really neat little tricks. One is you can do seed coating to introduce the microbe into your crop or into your orchard as a much better way to get it introduced, rather than having to spray where it's sitting there waiting for UV to kill it. So we're avoiding those external abiotic and biotic environmental effects that have been so detrimental to spray applications of biologicals. You can also have these additional benefits of having something like plant pathogen resistance, or accelerated seed emergence, or just better growth of the plant. Now, an example is, this is just some of my own work, one of my students, and we were just putting a very common insect pathogenic fungus, Bavaria bassiana, into Arabidopsis. We've done the same in maize, we've done the same in brassicas. Little caterpillar up on the side there is white because it's infected by this fungus. The fungus infects directly. Spore lands on, it goes inside, it grows in the insect and then sporulates. The leaf on the other side is Sclerotinia. What we found is, when we put this fungus inside a plant, and it quite happily goes inside a plant in Arabidopsis, it reduces the amount of Sclerotinia that occurs. It was a completely unexpected result, and a lot of labs have found this because this is an insect pathogen. The strain we use still kills insects, but it will give you that added benefit. The other area that we look at a lot more now is using multiple agents. Now, this was always thought of as just too expensive. You can't use two microbial products, but you can get some synergies by doing this, and the economics of it don't change because you've still got, you produce two products, but you're using them at half the rates. And those blue and red lines there represent what happened when we use two microbes independently added together, which is the red line, and then together at an equivalent dose. And you can see that we're getting almost twice the kill of diamondback moth using the two microbes. And so there's lots of possibilities around this. It hasn't been done a lot because it seems to be a complication, and the companies are relatively slow to want to go in this way, but it shows that the ecology of the organisms is going to matter quite a lot. So this brings us back to the older idea of integrated pest management. And this has been around forever. On the right, there's a couple of books I've been involved in the last few years. There's an awful lot of science around integrated pest management. Everything from using low doses of chemicals with biologicals to parasitoids with microbes, and it's becoming a much more rigorous science than it used to be. There's always been some really good examples out there of integrated pest management, but it tends to need that little bit at the bottom there, incentives to move to large scale use. The incentives generally have come from the grower organisations. In New Zealand, apples are produced under a thing called Kiwi Green, which is an integrated pest management approach that must be followed by the growers to sell the apples in certain places. And it integrates Bacillus Thuringiensis with some chemicals, with parasitoids. So there's a lot that can be done in this area when we start adopting the science together with the biopesticides, rather than just using a biopesticide as if it was your chemical equivalent. That brings us to the sort of growing area now, which is this whole microbiome. Microbes are everywhere. There's no plant that doesn't have microbes in it. There's no plant that doesn't have microbes on it or under it. The interaction of these microbes is what drives our health, and it drives the plant's ability to produce. It drives its ability to resist diseases and insects. So we don't have to rely on a single microbe, but we can apply into these environments and enhance the whole effect. So I won't go into this very much, but you can imagine if you're putting a biopesticide into soil, for example, for a control of a soil insect or a soil disease, and it is collaborating with the other microbes that are already there, you can get a beneficial effect. And they're the interactions that we can now study and understand how that happens. How do you get a stable population of your introduced microbe that will do jobs for you? So we know we can manipulate the microbiome for better pest control. Even things like the example down the bottom there is just growing companion plants under grapevines. It encourages the parasitoids, it gives them a longer life above ground. It improves the soil microbial communities. So this has been started, a bit of uptake around the world. There are companies, especially in the US and Europe now, that are trying to sell microbiomes. Now, there's a little bit of snake oil in this area. You buy a microbiome in a bottle, it's not necessarily going to be that useful. It'll be a whole lot of spore-forming bacteria because nothing else survives long enough. But the idea is starting to grow, and how we can understand things like sequencing the microbial community is actually really easy now. It's not technically difficult. And so we can start to look, and this is where I think some of the science is going to go. And then when you understand your microbiome, what do you have to add to get to a better position? I want to put up one example, and I apologize, it's a wheat example, but it's just one I knew of quite well. And just to show that you really don't have to lose if you decide to go a biological route. And this is from England, and there was a group of farmers, of growers, and some scientists and some industry people who got together and said, what would happen if we went completely biological in these monocultures, IPM, and just the standard chemical approach that we take? What happens after a few years? And there's a website if you're interested in this setting up on top there. But what they did was, and this has been going a few years, this is 2 different varieties, which is what the 2 columns are. And this is over the 3 sites around England. And what they've done is they've got the conventional, which is basically chemical control, IPM, which is a mix of chemical and biological, and then just biological. And what you can see is they really didn't lose production by going completely biological. Of course, it's a huge risk for a grower to go this way, because it's your money that you're putting at risk. But when it's done at this level, it can work with the right microbes. And these are relatively simple microbial products. This is not complicated stuff. So, of course, where the future might go, and I'll just put up one example, we've known how to manipulate microbes for many years, and we know how to make some of these microbial pathogens better at what they do. What hasn't been there is the public will for us to go this way, and it could be many years before that happens. But technically, we can increase the speed of kill. We can increase the virulence of these fungi. In this case, the ones I put up there, it's putting scorpion toxins into a fungus, so it expresses the scorpion toxin, and you can greatly increase the activity, and you can greatly decrease the time to kill, which is very important. And that's just one example. This is 2008. These papers were published. The science has come on a long way and can be done a lot better. But microbes modify themselves all the time, and we can do it. So where I've been going with this very, very brief talk is, in many ways, when we measure the effectiveness of a biopesticide, we have traditionally measured the wrong thing. We're not looking at things like plant growth promotion. We're not looking at multi-year sustainability. We're not looking at the improvements in your soil that you get from it. We haven't been really doing integrated pest management, although I'm sure all growers have a version of it that they have been doing. And this long-term sustainability from microbial community composition, I think, is really important. And never losing that what we're trying to do is make marketable products, foods. So I think that's all I had. This is also very much, I'm Australian by birth, so I'm just going to do a bit of cultural appropriation here, because the Maori have a very good view on this, in that everything is connected. In the Maori world, everything is connected, and sustainability is just how they think of the world and how they farm. So it's got a lot of resonance in this country. So hopefully that wasn't too long. Thank you.
Fantastic. No, thank you, Travis. That was a really excellent presentation, and we've got quite a few questions coming through the talk. So I'll pop a couple of those to you now. The first one from Rihanna was just asking how biopesticides can be effectively used in an open field condition as they are UV sensitive. Your comment on that?
Yeah, it's a really good question. So there are several approaches. One of the big changes that's come about is we're better at formulating, and a lot of it is around formulating. And I use the example because it was something that always shocked me that it would work, of locust control using metarhizium. Now, as an insect pathologist, metarhizium needs moisture to germinate and to penetrate. So the idea that it was being developed in sub-Sahara Africa for locust control, I just thought it was a joke when I first heard it. Not a very open-minded scientist, as you can tell. But they formulated it in oil drops, and the oil drops were enough to protect it and to allow the spore to germinate on the cuticle of in this very dry environments. And so that's a formulation trick. We didn't change the fungus, got the right strain, so it germinates relatively quickly, but that's not a particularly big trick. So there's a lot of simple things we can do. And really, chemical production has been doing this for years, because they've always used sunscreens and things in the formulations.
No, great. Thanks for the question, Rihanna, and excellent answer, Travis, as well. We've got another question from Andy, just asking if you could briefly explain why entomopathogenic fungi are said to be safe for beneficial insects in an IPM program. Yeah,
so generally when we are developing the safety, you've got to realize the context of it is dose. And so most of the entomopathogenic fungi we deal with are relatively specific to, say you're working on a coleoptera, they don't particularly like hymenoptera, which is what a lot of the parasitoids are, so they won't infect them. You can infect them if you hit them with a really high dose, and you can infect bees if you hit them with a really high dose, but it doesn't happen in the field because it doesn't get that level of dose, and they don't spend enough time in the zone where they get the dose. And so you could spray them with your formulated entomopathogenic fungus, and it won't kill them at a field dose. And it's something that we test for all the products that go out there, because it's an obvious one. But don't be fooled by the fact that it's safe, but it can still infect under some situations. The one area that you do see a little bit of antagonism is that you basically got two organisms competing for the same food source. And so they won't necessarily be happy that you won't get a parasitoid emerging out of a fungus infected cadaver.
No, thanks, Travis. Travis, one thing you mentioned throughout the presentation was the integrated approach and how that's been born out of other programs like IPM. Quite often the complexity of that approach is a barrier to adoption. Do you see similar hurdles here from a grower perspective, and perhaps one or two practical tools for those that are starting out in this area?
Yeah. One of the biggest mistakes that gets made in IPM is not to recognize what the microbe you're using is, and I've seen it quite a lot. If you spray a fungicide and you're trying to spread out an interpathogenic fungus, generally it won't go well. And so you've got to think about what are you integrating. There's no reason why you have to give up all your chemical controls. The idea is you're reducing their use, and the timing's important. So you can sequentially do things. So it's about, it comes back to understanding the science of what you're trying to do, and understanding how the pathogen that you're trying to use, say against insects, or what you're trying to use for disease control, when it works best and how it works best. But I've had some strange examples where we were trying to do work in onions. Now, onions are probably one of the worst crops for putting pesticides on. You can believe, like 17 to 25 different applications a year, and we were trying to put trichoderma onto them. Very hard to convince a grower not to do it. And we tried to tell them being a little bit pregnant was difficult, but they just can't afford to have failure. We were surprised that when we put trichoderma into the system, it actually worked. It did survive the chemical treatments. So it's not necessarily an all or nothing approach.
Nice. Thanks for that, Travis. One final question we had before we move on to our next speaker. You mentioned microbiomes and some science advancements there on the horizon in your tip. I'm just interested in your thoughts on time to market, because we mentioned that was a barrier, say, in the more conventional synthetic chemical groups. What is the time to market for some of these products?
Oh, it's good. It's going to be quite long. There are a number of companies doing it now, but they have some strange marketing models. There's one company in the States which actually buys the crop, guarantees to buy the crop after it. So it does the microbiome manipulation, and then it guarantees to buy it. It doesn't seem to me to be a sustainable model, and I think the science is still coming along. But I think the bit to look at there is that we're understanding more and more what happens when you add a microbe to these microbiomes and how to make it stable. And for me, that's where the science is going to advance first, and then the whole microbiome manipulation will come a bit later. But the pace of change is quite amazing. But I wouldn't sit there expecting it to be in crops, in vegetable crops or hort crops in the next 5 years.
Yeah. Okay. So we're working about 5 years plus.
Yeah, that's a very personal view, but yeah.
Yeah. Okay. No, good to know. Well, Travis, look, thank you very much for both of the presentation and those responses to the questions. Our next speaker is Professor Neena Mitter from the University of Queensland. And Neena's going to be presenting on the Australian researchers' perspective, now that we've heard the international perspective, on RNAi-based biopesticides, some recent innovations, and also covering a Bioclay case study. Now, Neena, I'm going to share your screen, and just while we're bringing that up, I'll give a short introduction, as I did with Travis. So, Professor Neena Mitter is the Director of Centre for Horticultural Science at the Queensland Alliance for Agriculture and Food Innovation within the University of Queensland, and has been involved in molecular biology and biotechnology in Australia and India for over 20 years. Her scientific journey began as an agricultural scientist in India, where she learned firsthand the significance of agriculture in shaping the world economically, socially, environmentally, and politically. So Neena, thank you very much for joining us today. I'll let you bring presenter view up, which we're very shortly going to see, and hand over to you to take us through your presentation.
Okay, thank you, Carl, and thanks for this wonderful opportunity, and thanks to everyone who has come to listen at this time of the evening. So thank you very much. Carl, can you see my slides? I am on slideshow.
We can, thank you.
Thank you very much. Thanks, Carl. So yeah, as Carl said, that I create ideas, and I love those new ideas, and then I also love making them or changing them into reality. So one of these ideas that I'm going to talk to you today was born in about 2012, 2013, and this is on using RNA-based biopesticides, and I'll slowly take you through it. And also, as we heard, it's also about Bioclay, a transformational biological alternative. I don't think so I need to spend any minute on this slide. We are all aware what a contemporary issue it is, and with the pandemic we are even much more aware of it now. I was shocked when I was reading that the people suffering from acute hunger was 135 million at the start of this year, and it's going to double by the end of this year, so it will be 265 million. So yes, it is an issue, and one of the ways, of course, as all of you are even more aware than me, is by using pesticides. So yes, we do need pesticides, and we do need to protect our crops from pests and diseases. However, it is difficult to develop new chemistries. You know, it takes about 13 years and $256 million in research to develop and register a new crop protection product. And maybe only 1 in 139,000 chemicals tested makes it from the lab to the farmer's field. So it's a big ask. But then we are all aware that we have these issues. You know, we are all worried about the development of resistance to pesticides. Of course, you know, our premium niche produce, we do not want any residue on it. We are also worried about runoff to our precious waterways. Lack of specificity, and I was hearing that in the questions asked during the earlier talk. We really want our pesticides just to kill the beast and not to kill our beautiful butterflies and moths and monarchs. And as I mentioned, it's hard to get those sort of safe new chemistries. So what today I'm going to talk to you about today, and yes, show off slide because University of Queensland Marketing Department took the slide and they want me to use it whenever I present. So this is about RNAi-based biopesticides, and where it's based on actually almost like nature versus nature. So here we have RNA as the biological active, and clay particles are just the fancy postman that are delivering this active. And I'll explain a little bit more on how the RNA functions, but the key point to note here is there is no residue. These can be specific, stable, sustainable, and safe. So the clay is pretty much a natural clay. RNA is from the pathogen itself that infects the crops. Now, what RNAi, actually, it earned its discoverer's Nobel Prize when it was discovered. So it's a huge leap in science. Very sort of in basic terms, I think I'm sure all of you maybe have watched CSI Miami and other episodes. We all know that we have DNA, which is double-stranded. Now, this double-stranded DNA actually forms a single strand of RNA, which is called the ribonucleic acid, and that RNA then forms a protein. And this is how everything is expressed, whether your fruit color, your vegetable flavors, etc. We need that single thread of RNA to make a protein. But sometimes what happens is, instead of forming a single thread of RNA, this forms what we call double-stranded RNA, or 2 threads of RNA. Now, when these 2 threads are formed within the plant, it's a very natural mechanism. They are formed, and when they are formed, plants are not multicultural. They don't like it, and what they do is they chop it up into small pieces, or they interfere with this RNA so that this RNA cannot form a protein. So this is what we use when we are generating genetically modified plants. So what we do is, if it is a virus or a fungus or an insect you would like to control using this RNAi, you take RNA or DNA from the pathogen and you make the plant express or make this double-stranded RNA within the plant system. And when the fungus attacks or a virus attacks, this double-stranded RNA actually interferes with the pathogen multiplication. However, as we all know, yes, it works beautifully well, but acceptance of GM plants still questionable. Hopefully it will happen. Concerns are there. Regulation of use. There is for me, it was also the cost and time involved, and you can't have GM for everything as well. It's hard to generate those GM plants also. So this is where this idea was born, that instead of making a genetically modified plant, if we know that double-stranded RNA is the active, can we just spray double-stranded RNA on a plant? Just spray it, not as just as you spray your fungicide or your insecticide. Will it work? And yes, it does. And this was the work done by Francis Stenlato in Spain. As you can see here, he sprayed the double-stranded RNA targeting this pepper mild mottle virus. Now this half is the unsprayed side and this half is the sprayed side. And yes, and then he challenged the leaf with the virus. And as you can see here, we have a protection from the virus. And then when I started looking at the literature, I found a number of papers with saying, yes, it works. You can spray the plants with double-stranded RNA and you will get protection. And I was really perplexed, you know, why isn't this being looked into or shouted, or, you know, that, wow, this is a beautiful mechanism. And when you read those papers, you realize that there are limitations, and very serious limitations of spraying. If you just make RNA, or a naked, as we call it, naked Dsrna, or just the RNA, and you spray it on the plant, it is very unstable. It will degrade because of enzymes on the leaf surface. It's not protected from UV and sunlight. Also, it can get easily washed away by rain. So in all these publications, when you reach the end of the paper, it was that protection window may be as small as 2 days or maximum 5 to 7 days. So this is not going to work, you know. This means that every 2 days, and Dsrna is not an easy in terms of cost, and I'll come to that. This was not a viable solution for the industry. So that's where the thought process happened, that can we somehow deliver this Dsrna, making it a stable application so that it does not get washed away by rain, it does not get degraded by UV or enzymes. It slowly gets released on the plant surface and provide protection. So this particular idea was first actually funded by Bill and Melinda Gates Foundation, and what we are using here are clay particles. And as you can see, James Bond in the slide. Now, this is my research fellow, and he likes it and does not let me take away from this slide. So what we are doing here is, we are using that RNA. It is the RNA from the pathogen itself. We put it on the clay particles, and then when we spray on the plants, we can address all those issues that I was talking about in terms of naked Dsrna. So here, just don't worry about too much about the heaviness of the science schematics, but if you look at it, it's just these clay particles which have this RNA. We put it on the leaf, and after that, it's really a natural mechanism. In the presence of moisture and carbon dioxide, this clay actually slowly starts releasing this RNA on the leaf surface. This RNA then enters into the leaf, and plant has the mechanism now to chop it up into pieces and wait for the pathogen to attack. And whenever that virus attacks or something, that virus or insect pest RNA will be degraded. So this is what we call bioclay. Bio is the biological active, and clay are the particles that carry this active. It can be applied as a spray application using your normal sprayers. So it is very much compatible with any technology being used at present to spray insecticides and fungicides. It is very specific. We can make it very specific because we are using the RNA from the pathogen itself, and we can make sure when we are designing it that it is
Sorry, Nina, I think we might have lost the connection there. Can you still hear us? I'm just going to take out your video. And Travis, you can still hear us. Neena, there we go. We've got you back.
It's restored, I think. Sorry. Thank you.
No, not a problem. We're using a bit of bandwidth, so back to you. Thank you.
No worries. As I was saying, this is it's clay layers. They degrade very naturally on the plant surface, and there is no residue. And of course, there is extended stability. And this provides. It says organizer has stopped sharing your webcam. Is that the case, or you can still see?
I have, Neena, just to free up a bit of bandwidth if it is having trouble, just so we can still hear and see your slides.
Okay, no worries.
Okay.
Yep, that's perfectly fine.
Thank you.
So as I was saying, it's almost like nature versus nature. You know, your plants anyway get infected with these pathogens, and they have the RNA of these pathogens in your plants. What we are trying to use is using that actually to kill the pathogen. And it works. It beautifully works. Once again, it's a little bit of a sciencey slide, but you can see, if you look at these big black blobs. Now, this is after 30 days of application of the Bioclay, and you can still detect that it is present on the leaf surface. If you spray it as a naked, you can see that these black blobs start disappearing. What you can see here is, if you spray just naked Dsrna and you wash the leaves, it's gone. But if you spray it as Bioclay, it just sticks to the leaf surface and provides protection. Similarly, this is just to show that it enters into the plant cells, but the best is this slide. So I wrote to Francis Tenlado and I said, could you give me the Dsrna that you sprayed on your plant when you published it in 2003, so that we can see whether we can improve it when we use it using Bioclay. And this is, yes, it is. Now, the plant was sprayed on day 0 and then challenged with the virus on day 20 instead of 3 or 5, 7 days. And you can see that it still protects. And we have gone ahead and it gives a longer protection than that time period. We have now progressed. We are testing number of viruses, and this particular work is being done with the Hort Innovation, Cotton Research Development Council, and New Farm Australia Limited as partners. And you can see here, this is the plant which is unsprayed, and this is the plant which has been sprayed with the clay and then challenged with the virus, and it provides beautiful protection. Not only viruses, we are looking at insect pests, and this is just to show we made the RNA fluoresce so that we can see it. And this is the fly which fed on a plant that was sprayed with RNA, and now it has gone into the gut of this white fly, so that we can target genes in the white fly so that the white fly multiplication can be controlled. Also, it is, because it's a beautiful platform, it not only works against viruses or insect pests, we are testing it against fungi. And as you can see here, this is a work done by our collaborator in University of California, Riverside, Professor Hailing Jin. And these are the plants which were sprayed, or not plants here. So this is tomato, strawberry, grapes. And best part about this slide is that this shows that we can even use it for post-harvest. Now, this is very early on stages, but it does give a little bit of an indication that if we treat these with the RNAi, we can get protection, whereas if these are just your control plants. And we have also done work on lettuce leaves and onion leaves and rose leaves, just to show that this clay actually, or the RNA, can provide protection. My battle with the thing was that when we started it, I was, in science terms, I was very convinced, yes, it's non-GM. Now, why I call it non-GM? We are just spraying the RNA. We are not integrating it into the genome of the plant. Anything is genetically modified only when you put it into the genome of the plant and it starts multiplying with the plant. In this case, it's like any other active. It will be only whatever we have supplied, and then after it finishes, it finishes. So it's, you know, you'll have to spray it again after whatever be your schedule. Maybe it lasts only for 8 weeks or 10 weeks or 12 weeks, and we are still working on that with different pathogens. But there is no genetic modification. There is no integration into the genome of the plant. And I'm very pleased that 2 years of my trips to Canberra, and Office of Gene Technology Regulator, OGTR, has legislated that yes, topically applied RNA is exempt from GMO regulations, and this was passed last year in November. So this is something very nice. And I'm also not only so in US, in Brazil, in India, I'm a panel member on Europe as well. And we are still sort of developing the guidelines for Europe to come to a decision. This work actually, which started in, as I mentioned, in 2013-14 with a team of 3, has really grown. So I was telling you about the Hort Innovation CRDC New Farm project, which is on viruses and insect pests of vegetable crops and cotton. And we also now have a big hub with about 50 partners in it, 4 universities, 3 state government departments, all the RDCs. So Hort Innovation, CRDC, GRDC, Wine Australia, Osveg, we have, as a peak body, we have all of them, and we are looking at controlling fungal diseases. Now this hub just started last month, so I thought I'll put that on. And in this hub, we are taking it all the way. So, you know, right from designing to the lab trials to the glass house, field scale up, registration. And we also are looking at adoption in terms of engagement with the consumers and growers to define the pathway of social licensing. So it's a work in progress, but yes, it's happening. So if I think of RNA-based biopesticides, we started as a spray, but we are looking at different applications now. Can we make plants, you know, put it in a tissue culture media and protect? Seedling applications, baits, or feed. We are at present, I'm also working on a project where we are looking at fire ants. So can we put it in the bait of fire ants, and when they feed, can we get a control through that? We have a project where we are looking from plants. Now we are also looking for animal health like sheep blowfly. So it has a bigger value, and yes, it has a value across plant and animal health. And as I say, for agriculture of tomorrow, I say yes, it has, you know, endless possibilities, you know, whether it is protected cropping, where, you know, it is very important that you control your maximum residue limits. And MRLs is a big issue with the export produce. To have Europe, I know, is almost sort of trying to move towards a chemical-free agriculture. So these are the kind of innovations then they can help. The only thing to keep in mind is, and as Travis was also talking, and I would echo that same sentiment, there is no one solution that can be, you know, the golden solution. We have to look at IPM. So when I'm looking at Bioclay, I'm also looking at as, you know, you have your pesticides, but this could be a wonderful additional tool. In some situations, it may be the only tool that can work, but in other situations, you may need you know, other interventions. So yeah, very excited that it's a UQ owned, University of Queensland owned and invented Bioclay, and we are looking at moving it towards the product development. So yeah, thank you from the Bioclay team, and happy to answer any questions. And I'm sure, you know, I know it is a little bit, it takes a while to warm up to this, but it's a wonderful innovation.
Thank you, Neena. Excellent presentation, and we've even overcome a bit of a hiccup with the National Broadband Network in between there as well, so thank you. I've just sent you a request to turn your webcam back on so we can see you for the questions, and we'll get stuck straight into those, given that we've only got 5 minutes left to go.
No worries.
We've got a question come through just asking, is Bioclay a systemic pesticide, as one of the slides showed it was actually entering the plant itself?
Very good question, and thanks for asking. Yes, it is. It is RNAi, like its RNA is systemic. It can move. So it can move down, it can move up, it can move through the vascular bundles of the plant. Only thing to keep in mind here is it's only a finite amount we are supplying. So whatever we are supplying, yes, it'll move. And we have done experiments where we have, challenged the new leaves that come out, because that was one of the question in my head. It's not only the sprayed leaves, viruses or insect pests comes on fresh new leaves, whether those new leaves will be protected, and the answer is yes.
Right, thank you. Another question that's come through, Neena, was just around the clay itself. Does that interfere with plant photosynthesis at all?
No, and you won't be able to even see this clay. And in fact, this particular clay, or what we call layered double hydroxide, actually is used as a slow release fertilizer in agriculture. So it's a very, very safe and no effect on photosynthesis or yield.
Okay, interesting to know. One other question that's come through, and thank you for sending those through during the presentation. That's really useful to go back and refer. So I've made a couple of notes here. We've got one question from Mike actually asking how many target double-strand RNAs can be included in the one spray, or is it one pathogen or insect per application or formulation?
Oh, my dream is, like if we have a fungus and an insect and a virus attacking a plant, I should be able to make the cocktail of Dsrna. It's possible. So we are exploring that, and you could have, multiple combinations.
Thanks for that question. One other from Danny, just saying, if the clay can be used in post-harvest, does it need to be washed off, just from a residue point of view?
Good question. And as I said, this post-harvest thing is very, very early stages, Danny. The thing with the clay is it's a naturally degrading clay, so it's just got magnesium in it. It just degrades in the presence of moisture, so it doesn't remain as clay, it just becomes those magnesium ions. But a good thing to look at. I must admit that this is something that we can investigate more.
Thank you. And perhaps a similar question in that we don't know, just in the early development phases, from Andy, just asking whether you can use a specific insect disease that is essentially broad spectrum, or is it species specific?
It depends upon, you can. So what we are trying to do here is, when I'll use the word broad spectrum, we put a family, if we select let's say whiteflies or any aphids, we can design RNA which can control different types of aphids, whether it's Mysis persicae or Apis gossypi or any other aphid, because what we can do is we can look at their DNA or genome and pick up sequences which are conserved, which are present in all of them. Then it will work against all of them. If we pick up only a specific one that, no, we don't want to target anything, we just want to target only this particular species of insect, then we can do that. So it has that kind of a flexibility.
Excellent. We've got time just for one last question from Jeff, just asking what are the main risks that you see with using RNAi products?
Once again, as I was mentioning, I was in Paris last year, and then this was totally about discussing the fate of Dsrna in the environment and any risks associated. Now, and of course, we need to deliberate more on it, but the message that comes out is that GM crops, they express a lot of Dsrna, and they're being consumed. We don't have them here, but yes, they are being consumed, and there's nothing that happens. In this case, the amount of Dsrna is minuscule. We are not even supplying, a great amount of it. And also, lots of studies have been done, and Fazan's has said this as they put it out, the Food Australia New Zealand labeling one, that Dsrna doesn't present any risk. In the human system, if you, let's say, ingest Dsrna, because it's been sprayed, you do that anyway, because sometimes you never know, whether you're eating an infected plant, and that may have the Dsrna. And Dsrna in the human gut, it doesn't survive. It just gets degraded pretty much you know, and people have done very large elaborative experiments on that. So it'll be case by case, but at present the verdict is that it'll be safe, but we need to watch out that space.
Thank you, Neena. Well, that has been an absolutely excellent and very fascinating 1 hour, and a great way to spend a Thursday afternoon if you're in Australia, or morning if you're over in Europe or elsewhere. So look, thank you very much to both of our presenters, Travis Clare and Neena Mitter. That has been a very prompt but very thorough walk through where we are up to in biopesticides research. Just a couple of pointers before we do finish up. Some resources and events that might interest you after this session can be found on the Soil Wealth ICP project website, and also those handouts that I mentioned earlier, a couple of those being the biological products database relevant to this session, and some other fact sheets. This session is being recorded too, so we'll have that available up online very shortly. But look, thank you very much for joining us. We've just come in on the hour, and we hopefully will see you at the next event. Thank you, Travis. Thank you, Neena, and we'll see you soon. Bye, everybody.
Thanks, everyone. Thank you.